Good day, everybody. Thanks for the opportunity to present something to you about management of Spongospora on potatoes in South Africa. Most of you know me. I was involved with potato research for many years, but from the beginning of 2022, I took up a job at Citrus Research International, so I've now moved on to citrus. So for those of you who are not familiar with Spongospora, although I'm sure most of you are, I would like to introduce you to the diseases that are caused by Spongospora on potatoes. So the first disease that we know that is caused by Spongospora is infection of the roots, and this obviously leads to disrupted root function. So what you can see in those feeder roots there is zoospores, which have infected the feeder roots, and therefore the roots are unable to take up water and nutrients as they should. The second symptom, which is visible to the naked eye, is root hyperplasia or the development of root galls. And this similarly leads to disrupted root function. So if one digs up infected plants and you examine the roots very carefully, you should be able to see these root galls on the fine feeder roots of the potato. And then, of course, the symptom that we all know well is powdery scab, which can be seen on tubers, and this leads to yield and quality losses. So whenever I give any talk about management of any pathogen, I take us back to the disease triangle or the disease pyramid. In order to manage a pathogen, we need to understand the interaction of factors that lead to development of disease. So essentially, we want to minimize the pathogen, as you can see in the bottom right-hand corner. We want to make the plant stronger, and we want to reduce the environmental conditions that are favorable for disease development. So how do we go about doing this? Well, it's also important to understand the disease cycle so that we know where the critical points are in managing any pathogen. So I always like to start with initial inoculum. And in the case of Spongospora, the initial inoculum is in the form of resting spores, which are found in cystosauri, also known as sporosauri. And this primary inoculum is very resilient to any kind of control measure and even to degradation in the soil, and can survive up to 20 or more years in the soil. These cystosauri also survive passage through the digestive tract of animals, which makes them highly resistant. And of course, this also poses a problem to growers who feed their contaminated tubers to animals which later graze on potato fields. So in some cases, some anecdotal evidence says that these cystosauri are able to survive up to 50 years, and that is very worrying. So where is this primary inoculum found? Well, it's found in contaminated soil and also on infected seed. And the question is, why are these cystosauri so highly resistant to all kinds of external stresses? Well, let's have a closer look at a single cystosaurus. If we zoom in, we will see that this cystosaurus, which is approximately the size of a grain of sand, is composed of smaller structures, and these smaller structures, as you can see here, are individual resting spores. And within each cystosaurus, there are anywhere between 700 and 2,000 resting spores. And then if we look again at an individual resting spore, each resting spore contains a zoospore. Now, I'm sure that most of you know already zoospores are infective structures of many pathogens, and they have flagella, which allows them to swim through any kind of liquid medium. So in the case of this pathogen, when a susceptible host is planted, the roots of the host release root exudates. And these root exudates form or serve as a chemical stimulus, which results in germination of the zoospores from the resting spores. And the zoospores then swim towards the roots and infect the roots. This process only takes about 15 minutes. So how do we lower the initial inoculum concentration in the soil or on seeds of Spongospora? Well, we first need to know where this is found and what the threats are of various different aspects of initial inoculum. So a couple of years ago, I visited some potato fields along with a colleague from New Zealand, and the grower there mentioned that root galls and powdery scab were detected on potatoes that were planted in virgin soil. And these potatoes that were planted were actually also mini tubers. So the question arose whether the pathogen is actually spread by wind. So we did some brief studies having a look at concentrations of the pathogen in virgin soil versus cultivated fields. And what we found was that in virgin soil, although the concentrations were low, we were able to detect Spongospora. And this essentially is a worry, because what this tells us is that Spongospora may be spread by wind, and therefore we essentially don't have any virgin fields that, if they're close to a potato field, that are entirely free from Spongospora. So one should always, if you're planting in a virgin field that is close to a potato field, you should consider it a medium risk, as the pathogen is windborne. The same study or a similar study was done on the Gaza Strip in Israel, where they have very similar conditions to the Sandveld, and the same results were found, that the pathogen can be dispersed by wind. There are certain methods that can be used to test the soil pre-planting to determine the levels of Spongospora in the soil. In further studies, we also had a look at various soil treatments that could possibly be used to suppress Spongospora. This is available in an article published in Potato Research. We found that the most promising are still the chemicals such as fluazinam and metamsodium. However, we do know that these are going to be removed from the market not too long from now due to the European Green Deal. We need to start focusing on other products that can be used. So one of those which also showed promise was calcium cyanamide, which is essentially registered as a fertilizer, but does have a biocidal effect. So this is one option, although I do realize that this is a very expensive product and not necessarily always that accessible. We also looked at various biocontrol products, those containing trichoderma and or bacillus, and they also showed products in the suppression of Spongospora when applied as soil treatments. So in follow-up studies, we did similar trials in fields over 3 years, and we looked at different chemical, biological, and soil amendments in 3 different regions in the country, and we found similar results. So just to show you the number of treatments that were applied in different field trials and the plot sizes, you can see it was quite an extensive study, this. I'm not going to belabor the specific different products that worked in each area. However, I would like to show you a summary of products that worked generally well for all of the field trials. So first of all, in terms of root gall management, the products that we found that had good suppression of root galls were calcium cyanamide as a pre-plant application in the soil for both powdery scab and root galling, Bacillus ameloliquefaciens as a seed treatment, both for root galling and powdery scab, fluazinam in furrow and dichlorofen in furrow, and then that suppressed root galling, and then trichoderma species also in furrow showed a good suppression of powdery scab. What we found that did not work so well was trichoderma as a seed tuber treatment and Bacillus ameloliquefaciens as a foliar treatment. What also has to be noted here is that some products performed well at low disease pressure, but did not show the same good results when the disease pressure is high. Therefore, it is critically important to know the disease pressure in the field when making product choices. So when we consider the disease cycle again, we need to bear in mind that this is actually a polycyclic pathogen, unlike most soil-borne pathogens, in the sense that it produces secondary inoculum in the form of zoospores. So these zoospores move in the soil water and can infect more plants. So what are the critical points in the disease cycle where we want to focus control measures, or at least we need to know what these points are? So the zoospores can infect young tubers, and the critical tuber infection period is just after tuber initiation, and this infection must occur by 40 days after emergence for one to see development of powdery scab. Most powdery scab is noted, however, when the infection occurs really early in the season. And infection of young tubers occurs between 9 and 17 degrees. In terms of the infection of root hair, this is a slightly different picture, where we see that infection of root hairs occurs right throughout the plant life cycle. And it begins approximately 2 weeks after root formation. So should one want to observe the development of zoosporangia in the roots, one should look anywhere from 2 weeks to 45 days after inoculation, or for root galling anywhere from 45 to 75 days after inoculation. And root infection occurs at a slightly warmer temperature range from 11 to about 25 degrees. So coming back to the disease triangle and looking at these critical infection periods, how can we minimize the favorable environmental conditions under which the pathogen infects potatoes? So when one looks at literature, it's not always easy to extrapolate the data from other countries to South African conditions, because obviously our conditions are different, cultivars are different, soil type is different. So we conducted some studies to have a look at the optimal temperature and soil moisture conditions for infection of potato roots by Spongospora. What we found was most infection of roots occurs when the temperatures are at approximately 15 degrees Celsius. Literature says 17, so it's pretty much the same. Above 30 or below 10 degrees Celsius, we do not see a lot of root infection. In terms of soil moisture, the earliest infection of roots was noted when soils were kept at saturation point. However, the greatest amount of total infection was noted under fluctuating soil moisture conditions. So what are the applications of these findings? Well, firstly, one should choose a planting date such that the temperatures during initial root development are greater than 20 degrees, because this will mean that there's not as much initial root infection. And secondly, irrigation scheduling should be used to avoid fluctuating soil moisture content in the soil. So bringing it back again to the disease pyramid, how can we strengthen the plant? Are there ways in which we can strengthen the plant to build up its resistance towards this pathogen? Well, here we have to look at the life cycle again. With Spongospora, there are two sections in the life cycle. The first is what we call a complete life cycle, and this occurs in primary hosts such as potatoes. And in this life cycle, the pathogen is able to produce cystospore again, thus increasing the soil inoculum. So we want to avoid these kind of primary hosts in any rotation cycle with potatoes. On the other hand, there is an incomplete life cycle in which no cystospore are produced due to something lacking in the plant that does not allow the pathogen to produce resting spores and cystospore after infection. So this life cycle does not increase the soil inoculum. And for that reason, one would like to include these non-hosts and trapping crops in a rotation cycle. So a number of studies have been done in my group, and one is continuing at the moment to look at alternative hosts of Spongospora in South Africa. And we first looked at crops that are commonly planted in South Africa. And you'll see this classification here where I show in orange crops that have been shown to be full hosts of Spongospora. So these kind of crops we want to avoid planting in a rotation cycle with potatoes. And unfortunately, these are a lot of the crops that are currently planted in rotation with potatoes. Those shown in blue and green are crops that can be planted in rotation with potatoes, as they are either non-hosts or trap crops. You'll see I do have a question mark after carrots and black oat, and this is because in some studies they've been shown to be hosts, while in others they were either non-hosts or trap crops. And I put this down to the use of different cultivars in different studies. So essentially, these studies need to be repeated using a wider range of cultivars. The same study was repeated with cover crops, and this is the ongoing study that we're looking at. So the use of cover cropping is definitely recommended for sustainable production of any crop and for improving the health of the soil. But unfortunately, the package of cover crops is not so easy to put together when we look at the host range of some of the cover crops. And this is specifically the case with Spongospora. So of the cover crops that we screened, the majority of them were also hosts to Spongospora. There were, however, a few trap crops as well. So this is slightly concerning. But what one also needs to remember is that we're not only dealing with one pathogen in this, we're dealing with a number of pathogens. And I just drew up a small table having a look at the host range of Spongospora, Streptomyces, and Rhizoctonia. And unfortunately, this also doesn't paint a great picture in terms of our selection of crops. But what this does tell us is that if one knows the history of a field and you know which pathogens are most prevalent in a specific field, then one can choose the crops planted in that rotation such that you do not choose hosts for the most problematic pathogen in that field. So crop rotation is also therefore very important, and to this end we have a long-term crop rotation trial running in the Eastern Free State with some colleagues where we're looking at different rotation strategies and sequences that will best benefit potato health and the health of other crops as well as soil health. So the other way in which we can try and manage Spongospora is the use of different cultivars and by using the plant's inherent susceptibility or resistance to play in our favor. So cultivar susceptibility trials were planted over 10 years in 4 different growing regions, and I basically just summarized the results briefly here. So what I'm showing here is cultivar susceptibility to root galling, firstly. You will notice that not all of the cultivars were planted in all of the trials, so I don't have a lot of stats for some of the cultivars. But generally what I want you to see is we do have a variety or a range of susceptibility in terms of root galling. Those cultivars highlighted in the red block are highly susceptible to root galling. Those in blue were moderately susceptible, and those shown in green, although not all of them were planted in more than one trial, did not develop a lot of root galls. So there is a choice in terms of root galling. A similar picture can be noted with powdery scab, where you will see that we have a range of susceptibility in terms of the cultivars that are planted in South Africa. Now you will notice that not all of the cultivars fall in the same category for root galling and powdery scab, and I will explain that very briefly in the next slide, or in the next couple of slides. What is also important to notice, however, is that although there is a choice, the question is, does the farmer really have a choice? Because if you look at this previous picture that I showed you, the tubers underneath the top picture there are the ones that are generally most resistant to powdery scab, but that's not the kind of tuber that our housewife wants. Our housewife wants perfectly waxy finished skin potatoes, and generally they are more susceptible to powdery scab. So why is there a difference between root and tuber susceptibility? Well, these differences result in or due to various reasons. First of all, the environmental conditions determine the susceptibility of the pathogen. There is a gene by environment interaction, which means that the environment plays a relatively large role in the expression of resistance. Also, the resistance expression is under different genes for root versus tuber susceptibility. Agricultural practices also play a role in terms of susceptibility of the pathogen to susceptibility of the host to the pathogen. The other thing is that Spongospora has a very patchy distribution in the field. So one could find that in some parts of the field there is a very low inoculum of Spongospora, and therefore one might not detect root infection, for example. The time of tuber initiation plays a role, and coinciding with that is obviously the environmental conditions at the time of tuber initiation. And this then links up with planting date and pathogen population in the field. So bringing it all back together, we want to have a look at what we can do to strengthen the plants again. So we need to have a look at the selection of fields, the preparation of fields, planting date, irrigation management, seed and soil treatment where necessary and where economically feasible, rotation cycle, and cultivar choice. So again, this is just to show you the different aspects of this management that can be used. So long rotations, a minimum of 3 to 5 years with alternative hosts, with non-hosts that are not hosts for Spongospora. Clean the seed or use clean seed and treat it if necessary. Treat the soil if necessary. Plant a tolerant cultivar if at all possible. Ensure that fields are well drained. Use irrigation management and schedule a planting date to ensure that environmental conditions are not favorable for the pathogen. Ensure proper nutrition of the plant, avoiding excess nitrogen. Excess nitrogen makes the plants what we call soft and more susceptible to infection by Spongospora. And then the correct choice of rotation crops, as I have mentioned. So some other important research that has been done on this pathogen in my research group is identification of differentially expressed genes in tolerant and susceptible potato cultivars and linking to that metabolomic fingerprinting of these cultivars, as well as the population biology of the pathogen in South Africa. And these results do have important implications for breeding and for management of Spongospora in South Africa. And you can get these articles from the journals if you are interested. And all of this research that I have presented today is available in various editions of CHIPS, which are all available on the Potato South Africa website. And then just some acknowledgements for funding over the years, Potato South Africa and the National Research Foundation. Students that have worked on these projects, obviously without students, I would not have been able to present this work. Various collaborators from Scotland, Israel, New Zealand, and Switzerland, and my admin assistant. And thank you for your time.